So good afternoon, everybody. Thanks for coming to this seminar. And I'm very delighted to have Alexis Hervais-Adelman here. Alexis, this is our first connection into the option to work and discuss things together. I was recommended Alexis from Professor Ivana Chenkova, who is the main professor of the interpreters at the Faculty of Arts. And I was curious how the interpreting is reflected in the human brain. And she recommended me Alexis. So that's why we have Alexis here. And I think it's very important for our field in general, because as the research is progressing and the language models seem to do all the things that the humans do with the language, I think we need to come back to the humans themselves and like connect the field also with neuroscience or neurolinguistics. So with that, Alexis, the floor is yours, and tell us everything, including your affiliation, because I'm not sure if you're in Geneva or Zurich.
Both. Okay. All right, great. It's a real pleasure to be here. I'm going to bore you with a little anecdote, which is that this is the second time I've come to this room. The first time was 12 years ago, maybe 13 years ago, in the context of this same series, talking about the very beginning of the work that I'm going to talk to you about the extension of. And it's a real honor, more than just a pleasure, a real honor to be here in my own right. That at that time, I was working with Professor Barbara Moser-Mercer, who some of you may know, at least by name and reputation, who, along with Nali Gulestani, was responsible for initiating a research program in Geneva on the neuroscience of interpreting. And it's thanks to them, I do my thanks at the beginning so that I don't forget, it's thanks to them that this is happening at all. And it's really nice to be back and talk about the fruits of what started 15 years ago in Geneva. The other people on this slide, this is a treasured collaborator from the University of Stockholm, Laura Babcock, and over here, a team in Turkey with whom I've been working on sign language interpreting. And these are the people without whom nothing could happen. So, you know, I'm going to talk, but everyone else is very important. So just back to the beginning for a second. I am at the University of Geneva. I'm also working at the University of Zurich, where I have a team where we don't work so much on interpreting. The work that I do there is related, but it's primarily about syntax, and I have a project looking at in utero development of language. Very, very different things. What unites my research interests in general is that it is language, and that I like to think a bit outside the box of what humans do with language. So Andre has invited me to talk to you about interpreting. I'm not an interpreter, I'm a cognitive neuroscientist. I'm going to try to justify myself. How dare I study interpreting from that perspective as best I can. If you want to ask questions throughout the talk, please don't hesitate. I'm going to do my best to keep the jargon low. I've included a section, I've prepared a section, which is really basic kind of neuroscience methods and stuff. If I see you looking very, very bored, or if you tell me this is boring, you feel free to tell me that I can skip it. And the point is hopefully to convey as much exciting and useful information to you all as I possibly can. And really the questions are a vital part of that. So I like to start with this rather silly joke. Here is a graffito. It says, "Life is pain." I am a bilingual speaker of French and English, and therefore I can enjoy this ridiculous pun of life is pain au chocolat. And me being able to do that, and you also being able to do that, requires, of course, that, oh, I see I'm going to have problems with the titles. No worries. We'll just have to guess what's happening there. Don't worry. Can I close it? If I can move my, yay. Super. Oh, yes. Thank you. Okay. So this little bit of humor that I hope has engaged you all. In order to be able to deal with the fact that this token, this bar or pain here, has two meanings, this tends to suggest that there are systems to which it is linked. And those systems for me are French and English. And of course, we can talk about how pain au chocolat has entered English as a term, but that's a very different conversation. I'd like to focus on the joke, if I may. And what, for me, is critical about this is that in order to do this switch between French and English. I'm talking about a switch, and switching is a mechanism, it's a cognitive mechanism. We always switch between tasks. You know, right now I'm giving you a talk. If a fire alarm went off, I'd be able to switch to emergency procedure and running or following your instructions. So what is it? What is it in the brain that enables us to do this? And of course, I'm very interested in that, and I need my mouse back, and I'm going to find it somehow. Ooh. Yes. So we have, and it is well known and has been discussed for a long time, there's such a thing as a multilingual language control circuit. And so this is a part of our cognitive apparatus which enables us to control the languages that we speak or that we use as multilingual speakers. It's potentially the case that we also use similar mechanisms when we switch register. Even as a monolingual speaker of a language, we are able to engage with people in different registers, to use a different vocabulary at home than we use at work, etc. That is far less well understood. And I'm going to stick to this notion of language control as being multilingual language control, the need to switch between the languages that we have. And you might want to ask me about dialects, and then I'm going to say, I don't know, I'm sorry. But it's also a very interesting question whether language control requirements are similar or very different if we're switching between dialects of a particular language that we speak. For instance, I'm very interested in the Swiss case, the Swiss-German diglossic context of, you know, is there such a big jump for the brain between Swiss German and High German for those native speakers? But anyway, I'm very much already going off piste. How do we get to the Bauer Chocolat? Some apparatus. Now this circuit, this is the naughty thing that you do when you go, yeah, I've spent years developing this, and I'm not going to talk about it in great detail. What I want to show here is that over years of working on interpreting, I've slowly put together a model that needs to be tested, which is a link between the neurobiology and the functions and the behavior that we've observed for control during interpreting that we think reflect the neural basis of interpreting itself. Now, I'm going to try and get you to roughly understanding this circuit by the end of the talk. This is not something to absorb right now, just to say that this is the fruit of a lot of work, and by the end of the talk, I hope you'll understand some of it, and you'll also hear how I'm not sure that I was right when I came up with this. And it's important to understand, or for me to tell you, that I question myself and that this model is a model and it has been slowly refined, and it may not be true or 100% correct. And that's why we're here, to improve it. So multilingualism is per se a very diverse experience. Different speakers use languages in different ways, and there are different contexts of use. So right now, I'm a multilingual individual, you all are too. We're using one language. This is a monolingual communicative context, even though there may be other ways. This has gone dark. I don't know why. This is surely my fault for fancy presentation. Yeah, it's not so readable, but let's hope. Okay, so single language context is one in which we have to maintain the goal of speaking a particular language, and we have to prevent interference from the languages that we're not using. There are dual language contexts of communication. Thank you. I'm sorry, everyone. Please don't fall asleep now. Dual language contexts, which are those where we know that our interlocutors, you know, we're speaking to one person over here and another over here, and this one might be a native speaker of Czech, and this one might be a non-speaker of Czech, and you might switch between them. And this is something that requires a different kind of control. You have to be very alert, in fact, to cues about different individuals. Who are they? What do they know? And you have to inhibit and select languages as a function of that in order to switch correctly. And I like to think of this as engaging or disengaging with a task. And this is something that I'll come back to, this notion that a given language or the other language, that these are different tasks in a way. It's a bit of a narrow way of conceiving of languages, but it's helpful when we think about the idea of switching, suppressing, inhibiting, because it's much easier, I think, to think of, you know, oh, well, you know, now I need to stop driving my car so that I can do something else. So it's task switching, than thinking about languages that are constantly interfering. So this is really just a cheat. I like to think of languages as tasks. You might agree or not, that's okay. Another way that people use multiple languages is in a dense code switching context. So this is a construct where you know that your interlocutor speaks more than one language, the same one, two, or the same two or three maybe as you do. And then you select to use terms or phrases or idioms from that other language, which is not the one that is the carrier language in your discourse. And you can just do this, and this is a very effective mode of communication. And this is something that's actually very complex because it requires continuous awareness of when you can do this, like particularly for mapping the morphosyntax of the inserted language into the carrier language. And of course, there's a dose of opportunistic planning. When is it good to do this? How am I going to do it? So the multilingual experience here, and this is, this is a non-specialist experiences. Most people who are multilingual will use languages in all these ways, puts pressure on a set of cognitive control systems that are quite diverse. And this is something that people have studied, people have thought about, and there's a huge amount of research on this going back 40, 50 years. And the most recent models here that I'm presenting from Calabria and colleagues have really been evolved by David Green and Jubin Abutalebi, who've thought about this a lot. David Green has been writing about multilingual language control since the 80s. Jubin Abutalebi is a clinician who's looked at bilingual aphasia, which is a phenomenon where people who speak more than one language have a brain injury and then suddenly show different mastery of their languages or inability to control which language they speak. And this kind of evidence, brain lesions that cause problems for language control, has been quite influential in developing these models, as has more experimental work looking at brain imaging studies, so without the need for brain damage. And this model here, which might look a little bit familiar because it's what I based my own on, is one where they try to highlight the subcomponents of the brain systems or those subsystems in the brain that are relevant to a single language context for an individual. So here they're really putting the accent on this competitive suppression network. And here for dense code switching, they're looking at what they consider to be the most critical, the opportunistic planning part of that. And this, again, I'm showing you these complex diagrams, not so that I have to talk about them and explain them in detail, but just to say there is a lot of work. There's a lot of thoughts. People understand some of what's going on in the brain for multilingual language control. These are really interesting in themselves, but I don't think you're an audience that wants to know about, what does this connection do? I mean, if you are, please, hands up and I'll do my best to explain. But what I'm interested in doing, or have been interested in doing, is expanding on this for another context, which is simultaneous interpreting. This is another multilingual context. It might not be an everyday context for most people, but it is nevertheless something that our models of multilingual control have to be able to account for. If it's possible, if the brain can do it, then we need to explain how it is possible. Interesting. It's very adaptive and okay. All right, so by the next sort of 10 slides, I'll be done. I'm sorry, everyone. So my take on simultaneous interpreting is that it's a particularly fascinating multilingual context because it is quite extreme. It is unusual, that's one extremity, but where it's really different. Oh, no, okay, sorry. Where it's, for me, quite extreme is that in order to do it, you have to maintain two languages alive at the same time. They have to be accessible. You have to have an ear on the incoming language and attention to your own output. And in order to communicate within your own mind from one language to another, both have to be active. And this is categorically different to a more classical case of switching between languages in a sort of punctate manner, which is what we would choose to do if I now suddenly needed to address someone else in another language, or if you needed to address one another in a different language to English. So this is totally different. And therefore, as a cognitive neuroscientist, it provides for me a window into multilingualism in action, as opposed to those moments where language changes. And this is really the neural basis of juggling between languages. They're alive, they're being kept up. They're both languages in the system, the source language, the target language, have to be accessible somehow. And so, as I said, any neurobiological theory of language control has to be able to account for simultaneous interpreting. In order to make interpreting tractable, I have engaged in a very, very deconstructivist approach. And this idea of cognitive decomposition is one that some scholars of interpreting repudiate fully, and I am completely sympathetic to that. Breaking down a task as complex as interpreting into components, which is what I'm going to do for the next slides, is an operational necessity, but it does not allow us to really account for one particular feature of how any task is done, in particular interpreting for the synergies between the parts. So I am not going to be able to tell you anything about the complex interactions between the components because my approach is to try to individuate those components. So this is something that I want you to bear in mind. Feel free to criticize. I criticize it all the time. It is a valid point of criticism, absolutely. However, this is a constraint of trying to operationalize the task, and hopefully in the future we can do better. So simultaneous interpreting, which I'm going to refer to as SI in the slides a lot, is a task that fundamentally consists of converting a target message, or rather a source message, into a target message, and by doing something that is a bit more than putting a big red arrow in. So if we just break it down, please follow me, even if you hate this model. This is my model, it's the best I can do. There's a source message, it is perceived by our sensory apparatus. This then makes contact with working memory processes. It is stored to some extent in the incoming stream. There is then this magical operation of linguistic processes, which I think, is pretty black boxy, but what I'm talking about here is whatever we think of when we think of comprehension. This is associated with our world knowledge, with semantics. This has a huge influence on what we can understand and how we understand it. There is then the need to plan an output, motor planning and articulatory planning. The motor system is then responsible for executing the production of a translated message. That itself is then perceived. We hear ourselves when we speak. And there is self-monitoring. We know that interpreters don't simply speak and never correct themselves when they make an error. They correct themselves. Therefore, we know that they are attending to themselves, to their output, and that there is a mechanism of attention and self-correction. And at the heart of all these processes, I put this big red dot of control, which I think of as being divided into the attentional control, listening to the source, listening to yourself, the motor control, which is a bit more straightforward, producing the correct stream of sounds, and the language control, which I think of as being really the most challenging part of this, the maintaining two lexicosemantic sets alive at the same time in order to enable this mapping from source onto target. And of course, this is very, very basic, and it doesn't account for several things, which we can talk about in great detail, like the language asymmetry, what's the difference between the source and the target language, like the expertise of the interpreter, and many other features, like, what is the content? Is world knowledge really relevant? Or is this something where it's entirely novel and they don't have any references? The weightings of these tasks this is arbitrary. This is just a statement of what I think is important. It is not the be-all and end-all. This is just the way that I've found to try to put the parts list together. And of course, my goal as a cognitive neuroscientist is to try to fit this into the brain. And this, unfortunately, doesn't happen by magic. I have to do experiments, and I have to think of ways of putting that together. And this is where I could switch to a bit of background on neuroscience and methods in neuroscience, and I will do that very briefly then. I'll begin with some ancient history of the 19th century, when phrenology was a fun thing to talk about because it makes lovely pictures. So phrenology was a notion that we have organelles in our brains, or rather the brain is constituted of organelles, each of which has a particular function. And those functions can be individuated and can be described, and consequently, their configuration would tell you something about the capabilities and personality of an individual, and this would be manifest in the shape of their skull. So the personality and cognitive capacities are reflected by the bumps on the skull. And although this is not something we believe in today, at least the spirit is in the right place. It is in our brains and in our heads that we place these capacities these days. And these were very elaborate models. There were many, many capabilities and capacities that were specified, that had loci in the brain. And so this notion that there's a particular place in the brain where an organelle is found, or rather a locus in the skull where an organelle is for this, for that, is something that goes back a long way. I mean, before even the 19th century. But I just want to point out that language in this particular model was found here. We don't believe that to be true anymore. The eyes are for vision primarily, and we put language more in the brain. But how did we get from this notion to where we are today? I can't give you the whole history, but I want to just support a little bit this notion of locus and function. So alongside all this phrenology that we do find slightly laughable, people were looking at the structure of the brain, and Santiago Ramón y Cajal was a microscopist who drew exquisitely detailed pictures of the cellular organization of the brain. And he identified different cell types, and he tried to explain how these cell types related to brain function. And this is something that we still ponder today. We still think about, why does this cell with its ramifications like this organize in this particular way? What can it do that this cell, which is star-shaped, can't do, and vice versa? Why are they organized in different places, in different layers? And this sort of microanatomy of the brain is something that's been a source of wonder and fascination, and we slowly understand microcircuits in the brain more and more. And it's thanks to visionaries like Ramón y Cajal that we ever got as far as we got. Because something else that came out of looking at the microscopes was something that Corbinian-Brodmann came up with, which is the idea that not only are there different cell types, but they are organized in the brain in relatively homogeneous patches. So these different cell types, they're not randomly organized. There are bits of the brain where there's more of this with this percentage of that, etc., etc. And he drew a cartography of the brain based on the homogeneity of the cells in the cortex. So just in case sorry, I haven't oriented anyone, but this is the front of the brain. This is a medial view. It's a section taken vertically through the middle of the head. This is the back of the head here. This would be the bottom and the top. Here we're looking at the middle, and here we're looking at the lateral surface. So if I were to turn around, it would be this surface. So these patches, these dots, these striations, these are his decompositions of areas in the brain that under the microscope look homogeneous. And what's really interesting about this is that with this cellular homogeneity comes a kind of functional homogeneity. This organization, these areas, these labels, these numbers are the designation Brodmann areas. And we still talk about Brodmann's areas today. I will talk with my colleagues about areas 1, 2, 3, 4, 5, 6, 22. This is still in use. This was a huge innovation. And what we understand is that there is indeed a functional organization that relates to the cellular organization. And this is something that became much clearer. So just a quick content warning. There are 2 lesioned brains on the following slide. They're not gory, but they are real. So please be aware of that. So this is something, this organization, this idea there of functional homotopes, and this is something that became very, very clear when neuropsychologists started gaining insights from brain damage in the 19th century. So these are this sample is a very important sample. This is just an illustration of another, but this is the brain of someone very, very important in the history of neurology and neuroscience. And that is a patient who was identified by this man, Pierre-Paul Broca, at the Hôpital Bicêtre in Paris. He encountered a patient while working there who was 50 years old. His name is Monsieur Le Borgne, and Monsieur Le Borgne could only produce the utterance ton, ton, ton. That's all he could utter. However, he had previously been able to speak prior to his admission to the hospital, and he was perfectly capable of comprehending instructions. So he didn't not have a language faculty, yet he had this problem with articulation. And after his death, Broca was able to get hold of, I don't know how legitimately, his brain. This brain is now in a museum in Paris. And he identified this abnormality here, which I think is really clear to everyone, that this hole looks unusual. And this here is a lesion in what's called the left inferior frontal gyrus, and it is now an area that bears Broca's name. So people often talk about Broca's area. And the conclusion that has arisen is that the syndrome identified by Broca's aphasia. So aphasia is generically a speech problem. It can be production or comprehension or other. But Broca's aphasia is characterized, like the patient Don, by non-fluent speech and spared comprehension. And this division of labor, if you like, or this separation of the language faculty into production and perception is something that is quite crucial to our understanding of the brain networks of speech and language. So the conclusion that we have here is that there is a functional organization, there is a distinction between production and perception, and that the left inferior frontal gyrus, this lesioned area here, is responsible for production. So this is very interesting evidence for a very specific part of the brain having quite a clear and specific function. Now, Broca doesn't record everything else that might have been wrong with the patient. His most flagrant deficit was that he couldn't articulate anything beyond ton. We don't know more. Maybe he had other deficits. But we do know to this day that lesions in this area tend to create a problem or produce a problem with speech production. So that is something that is held up since then. And then alongside Broca, but rather contemporaneous with Broca, but not alongside, in Germany, Karl Wernicke was working on aphasia in general. He observed many more than one patient. He found that there were other kinds of aphasia. He catalogued many symptoms. And what he particularly highlighted is the inverse case, patients who can speak fluently, but who don't understand. And this is a problem that's produced when there is a lesion in a very different area. So Broca's area is over here. So we're now posterior, further to the back of the brain, still in the left hemisphere, in an area that's now called Wernicke's area, but it's ill-specified. It's the posterior superior temporal gyrus. And this area, lesions in this area, are characterized by a syndrome where there is a loss of comprehension, there's fluent speech. Very often, however, that speech is incoherent, as though the patient doesn't understand themselves and doesn't correct themselves, and they often produce jargon and a wonderfully named word salad, and they switch words occasionally. And it's unclear precisely why that's happening, but it's very clearly to do with the fact that they don't hear themselves and they're unable to monitor correctly what they're producing. So the conclusion of this is just that there is an area responsible for comprehension, or at least heavily implicated in comprehension. So these two, Wernicke and Broca, they're still around. We still talk about them. There's some arguments for changing the names and sort of being more precise, but in the common vernacular, let's say they're always there, and I will probably mention them later on. So these are ways that we started to understand, or I wasn't there, but that humanity has started to understand, that scientists have started to understand the relationship between brain and behavior in language. But of course, we can't rely only on lesioned brains. If I were to try to study interpreting by only looking at the brains of interpreters who had suffered a brain injury, this would take quite a long time, and we wouldn't get very far. Unfortunately, thanks to huge developments of technology, we can do things that are non-invasive. So the history of non-invasive examinations of the brain really goes back not so far. So this guy, Hans Berger, who discovered using this apparatus, which is essentially a huge amplifier, that the human brain emits electrical signals that can be measured if you put little silver needles onto the scalp of one of your children. So he experimented quite a lot on his daughter and his son, which was completely acceptable 100 years ago. Not so much anymore. Yes, it was known, so that Galvani had obviously looked at electrical signals and muscles and jumping frogs' legs already in the 18th century, but this was the first time that a signal had been identified from the human brain that was meaningful, functionally meaningful. And what's really, the big step that Berger made is that he identified a signal, and this is one of his original recordings. This lower line is a time code. It's just a sinusoidal time code. And this is the recording of the electrical signal, the potential difference between two of the electrodes on a scalp. And so this signal here, you can see that it has structure. There's this here, sort of sinusoidal, then noise, 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 sinusoidal, noise, noise, noise. And what is remarkable about this is that this isn't random. This is actually periods where the person being recorded had their eyes open and eyes closed. And this was the first time that someone had the insight that these waves coming out of the brain actually had a functional meaning. And this wave, which is sometimes referred to as Berger's wave, but is more often referred to as the alpha wave, is a signal that we still find. We find it all the time, and it's associated with attention. When you have your eyes closed, you typically will be recorded with an EEG that's got very high alpha. If your eyes open, there'll be less of it. And it's a signal that we know is actually predictive of people's capability of detecting stimuli that are at threshold. So higher alpha means that you're less attentive. Lower alpha means you're more awake and alert. And this is quite a discovery. So that's 100 years ago. This was the first time anyone had put together a brain signal with function. So this was a huge development. So EEG, electroencephalography, is a technique that is widely used. It does suffer from a drawback that is going to be very, very relevant. And when I tell you it records electrical signals, and when we speak, our muscles create huge electrical discharges that totally mask the brain's signal in EEG, you will intuit that it's not going to be the most helpful technique for looking at interpreting an action. If someone is speaking, the EEG signal is very, very hard to make out because the musculature from the face and the articulators create huge, huge signals, much bigger, hundreds of times bigger than the signal we can record from the brain. So it's a very useful technique for doing certain things, not great when people are speaking. So there we have to think of other things. Unfortunately, time and amazing insights have given us other technologies for looking at the brain. And one of the ones I'll talk about the most today is magnetic resonance imaging. And this is a technique that allows us to look at both brain structure and function in a totally non-invasive way. It also has drawbacks when people are speaking, but I'll get to those later. And of course, I've chosen this very, very fancy spaceship-like image just to, wow everyone. But it is really a wow kind of technique. It allows us to make wonderful pictures of the structure of the brain. We can see really here, for example, the distinction between the white matter and the gray matter and deep brain nuclei. And this is something we can do thanks to the invention of this technology. And here we have a brain in its great glory. And MRI is what allows us to do this. And structural MRI, so the ability to look at brain structure in more detail, has also led to a kind of study that we couldn't have done before, which is to try to associate brain structure with either behavior or with expertise. So what are the consequences for the brain of having a particular skill or having learned to do something? Does that cause changes to the structure of the brain, or can brain structure predict abilities? And people have gone into this in great detail over the last 30 years with the availability of brain scans. And you might be familiar with a study about the London taxi drivers, for instance. So this was a study from nearly 25 years ago, which showed that a particular part of the brain, the hippocampus, which is associated with the storage of information, with memory, is expanded compared to other people in taxi drivers in London who have to learn the whole of the street map of London. So people can do this. They can do studies where they associate structure with function. Now, this is less interesting. This is just orientation in the brain. The brain has many features that allow us to make up labels. And I just want to make sure that everyone is relatively on board with, this is the left, this is the right, that's the top, and this is the bottom. Because I will show pictures of brains when I come to my results, and it's better not to be completely confused. I will always try to reorient you, but just these are some views of the brain. They'll come up a bit again and again. So there are many landmarks, and we use these landmarks for describing the brain. So we can see there's this longitudinal fissure. The brain is more or less symmetrical about that. And a lot of what I will talk about is in the left hemisphere of the brain, because this is the one that is dominant for language. So most of the time, people talk about regions of the brain that are specific to language or specialized for language. These tend to be in the left hemisphere, Broca's area here, Wernicke's area over here. The right hemisphere is also responsive to language, but it is less responsive. So both hemispheres do stuff, but the left is the one that's dominant for language. We've also got another landmark here, which is the Sylvian fissure, which separates the brain into the more dorsal, so upper, parietal and frontal lobes, and the temporal lobe here. And the Sylvian fissure comes up in language quite a lot because a lot of the areas that are heavily implicated in language are said to be perisylvian. They are around the Sylvian fissure. So I might talk about that if I lose my grip on the jargon and I let all the jargon out. If I talk about perisylvian, it's because it's around this area. There's also another very significant landmark, which is the central sulcus. The central sulcus is the deepest fissure in the brain in a way, in the cortex. It separates the sensory, so the somatosensory cortex from the movement, from the motor cortex, and is also the division between the frontal lobe and the parietal lobe. And the reason that the frontal lobe is important, I guess, is because it's that which is associated with the more complex, the most complex cognitive actions that we do. It's the part of the brain that is most expanded compared to our last common ancestor with the chimpanzees, or we assume it to be the most expanded. And in a sense, it's that part of the brain which is the most uniquely human of any of the mammalian brains. And the evolution of it's a very interesting thing, but that's not the point today. Here at the bottom of the brain, we have the brain stem. That's how the brain connects to the spine and to the rest of the body. And here we have a really interesting structure, the cerebellum, the little brain. And the cerebellum has as many cells in it, as many neurons in it as the rest of the brain, even though it's so small. And it's a very, very ill-comprehended structure. It's known to be essential for movement control and learning about movement, fine motor actions. So for instance, really learning to play a symphony on the piano, the cerebellum is heavily, heavily implicated. It's involved in many, many important visceral and vestibular circuits for balance and for automatic control and not falling over. It's a very interesting structure because it also has important roles in language that we don't fully understand. So there's some motor things, but there's also potentially some syntactic roles. There's also some roles that are associated with prediction of timing when we're listening to people. And I found it to have roles when listening to speech, which is acoustically challenging to understand. It's a very interesting structure. We'll see it a little bit later. We don't know everything about it by any means. It's fascinating. And it's been ignored for a long time because you don't see signals from it in EEG, and people didn't have any way of accessing its action. Now, I mentioned gray and white matter before, and that's quite important just to remember the brain here. This walnutty surface, the cortex, is a shell, essentially, and it's got a gray hue, and that grayness comes because it's full of the cell bodies of neurons. Underneath the cortex, the shell, there's white matter, and the white matter is essentially wiring. It is cables that connect brain areas locally and distally to one another. And we also have, using MRI, a way of looking at those in vivo, and we can generate maps of the brain's wiring diagram, if you like. These brightly colored, primary colored spaghetti here are the major fiber bundles that connect the brain together. The different colors refer to the principal principal directions in which they are moving. So the red is horizontal into the screen, the green is forwards and backwards, and the blue is vertical. And this video shows what's known as an in vivo dissection, where I've taken out a particular set of tracts from the whole brain, which are particularly relevant for language because they are the ones that connect the auditory areas with Wernicke's area and Broca's area. So there's this nice thing called the arcuate fasciculus, this stream of tracts that connect these very language relevant areas. And this is, again, it's a perisylvian tract. So this is why this term comes up. So this technique allows us to look at the physical connectivity of the brain without having to dissect it. And the connectivity, as you can see, it's a beautiful structure. But if you can imagine that we have these different areas that are responsible for different functions, and obviously connecting them is critical. If the brain were just operating with isolated functions, we wouldn't be able to do anything. So there is something about connectivity that is crucial to function. I won't be able to get into it today because it still eludes me. I worked for a full year and a half on looking at connectivity and simultaneous interpreting. I got nowhere. There's a lot to do, and I hope one day to bring this closer to completion. So this is structure. So we've had connectivity, this notion of white matter, the gray matter, convolutions, and how those might predict function. And then we have a last thing we can do with MRI, which is to look at function. And the function we look at with MRI, functional magnetic resonance imaging, we look at the proxy for nerve impulses in a way. So we think of neurons as having to fire in order to do calculations, in order to represent, in order to convert information. And we can't image that activity directly, but what we can look at is the metabolic activity that's associated with them. And what we see here is the result of analyzing the blood flow in the brain when here a participant on the top line has been presented with an image that is in their left visual field or their right visual field. You'll notice that the left visual field presentation activates the right hemisphere and vice versa for the right visual hemisphere presentation. And these areas at the back of the brain, these are known as the visual cortices. They're in the occipital lobe. And this primary visual activation is what we expect when someone sees something very, very simple, a flash of light, a checkerboard, for example. And this is just the brain's first response. This is the first place that visual information arrives in the cortex. And from there, it will be amenable to further treatment, further processing by other parts of the brain. And here with the auditory stimulation, here I've cheated. It's not just a simple auditory stimulus, it was words. And we see this large activation here, this is bilateral auditory cortical activation. So the auditory cortices behind the ears, although they're not directly wired into the ears. And here these perisylvian language areas, a Broca's area here, and a motor area which is associated with speech production here. These images are produced by looking at blood flow. They're only proxies for neural activity. So it's the best we can do at this time, non-invasively. In order to look at neurons firing directly, we'd have to go in with electrodes into the brain, which is something that very rarely happens. You can do it in a clinical setting for people who are suffering from intractable epilepsy. When it's pharmacologically intractable, then people want to try and identify exactly where the source of the epileptic focus is. And one way of doing that is to actually go into the brain and place electrodes in the brain and record, monitoring the participant or the patient continuously, potentially for many days, and eventually isolating what part of the brain is responsible for the epileptic seizures that they suffer. During that time, participants, as they are patients, are often very, very bored. And being bored, they are amenable to taking part in experiments, and you can ask them, would you like to listen to some words? Would you like to look at some pictures? And a huge amount of data can be generated with great precision, but we typically don't have interpreters suffering from pharmacologically intractable epilepsy whom we could ask to participate in a study. And therefore, that technique is one that I'll put aside for now. Okay, so this was a lot of fluff. I hope that you're a bit oriented in the brain, that the critical point is I'm going to talk a lot about MRI, functional MRI and structural MRI. And I'm just going to try and put this into the context of interpreting finally. So that was a lot. Thank you for your patience. Let's get to the meat of this now. I apologize for that spelling error. Okay, so we're going back now a little bit in time. We're going back. Berger and his EEG 100 years ago. This was the first time, as I said, this is the first time that anyone had identified functional signals that really could tell us about how the brain's operating and when. It took a long time, it took nearly 70 years before anyone used this technique in the study of interpreting. And this was absolutely groundbreaking work published in 1993 by Pecher and colleagues, who were the first to actually look at bilingual speech management. And this was, I mean, this is a huge development. Finally, someone had actually chosen to spend the time to look at the brain during interpreting. And they looked only at a few participants, and they produced these maps of coherence between electrodes. So the EEG, they had a few electrodes, so you can see the positions of the electrodes on a sort of map of the human head with the nose there. And what these diagrams show is essentially the signal broken down into frequencies and looking at the coherence between electrodes. So a kind of surface level connectivity, which isn't really how the brain is connected, but it's developing a picture of how the underlying brain is operating during various tasks. So this is an English to German and German to English translation or interpreting task. And this is shadowing in German, shadowing in English. And then there was a music and a maths task. And although it's very hard to genuinely draw conclusions about what is going on in the brain from these data, what is extremely interesting, at least, is that during these, in this band, during these quite speech production level tasks, there's activity which is, I mean, I'm going to ask you to use the eye of faith, which is a bit more left lateralized in these shadowing conditions and more bilateral in the interpreting conditions, suggesting a slight difference in recruitment of brain areas, but we're a long way still from fully being able to see what's going on in the brain. I always like to cite this work because I marvel that it was ever done because it was so hard to do back then. And I think this is great, but it asks a lot of questions. Like, we don't even know how really to interpret these data. It comes from a tradition of looking at scalp-based coherence maps that Pecci was really a progenitor of, but you know, that's as much interpretation as I can do. It basically tells us slightly different things are happening in the brain when people do different tasks. Great. From there, at least we know we're onto a good track if we want to look at and investigate interpreting a bit more thoroughly. So the next shift that really allows us to look at inside the brain, this is a technique that I like to introduce with this wonderful picture. This is positron emission tomography. I don't want to talk about it too much other than to say it requires an injection of radioactive tracer into the participant, which is then something that can be metabolized, or rather it shows where there is more metabolism. And as it breaks down, as the radiation is emitted, sensors like these pick up on the annihilation events that are going on and can sense basically where there is more radiation and less radiation. And not all PET scanners look like this. This is a beautiful one from the 50s. They're a bit more sane now, I guess. But moving on, using this technique, Klein and colleagues actually did a study. You know, this is before MRI was really accessible. Did a study asking people to do a translation task, not an interpreting task, a translation task in a PET scanner. And what these maps show, and they're always very dark, they're very dark in the original paper, I'm sorry, but what they show is the contrast between translating and repeating in L1 and L2. And this really is a picture of what's going on in the brain during a multilingual task. This is really one of the first images that's relevant. And what we see over here is areas close to and around Broca's area. Also in this L2 case, and a deep structure here, which is the caudate nucleus, which is part of the basal ganglia, which I'm going to talk about a lot in future. But just to note that this is a translation task. It was single word translation. So just hear a word and repeat. There was nothing sort of ongoing about this. Okay. So these tasks, intriguing. We can start to see a brain network that's implicated in second language processing versus first language processing. But we're far from interpreting. And then only a few years later, thankfully, can I get there? Yes. Years later, also using PET. This, again, absolutely groundbreaking study by Rinne and colleagues in Finland, studied the brains of 8 simultaneous interpreters, very experienced professionals, and looked at the pattern of brain activation using this same technique while they did an interpreting task, and compared while they interpreted from English to Finnish and Finnish to English, so the habitual and less habitual direction. And they generated these lovely maps. And so to understand these maps, these are what's called glass brains. So you're seeing through the brain. This is a view from the top with the right hemisphere, left hemisphere, front, back. This is a view from the side, the front, the back, top, bottom. And this is a view from the front, the right hemisphere, left hemisphere. So in theory, you should be able to find any of these patches in 3D, but it requires quite a lot of brain reconstruction. What this task was quite simple interpreting, but nevertheless, it shows something very interesting. It shows two major activations, one in what's basically Broca's area, so this speech production area, and something that's more in the midline of the brain, in the supplementary motor area, which is an area that's associated with motor planning, but not specifically language. And down here, we also see something in the cerebellum, which is quite interesting to see in a task for interpreting, because as I said, the cerebellum is known mostly for its motor responses. And this last row is the difference between these two maps, the significant differences, and the significant difference between interpreting in the habitual and the non-habitual direction appears to be focused around Broca's area, so in the speech production areas. And then people did move on to MRI finally, and this is just a lovely picture of the first clinically useful MRI scanner. I showed you a really spaceship-like modern one. This is the first one from 1980. Very, very clunky. But Ahrens and colleagues, using a slightly more sophisticated scanner, finally did an MRI study looking for brain activation during an interpreting task, during a simultaneous interpreting task. This study never came out by itself. It's a chapter in a book, and that explains why I have a terrible photocopy, and I'm sorry for that. But what they're showing is left perisylvian language areas, a little bit of motor cortex, and some right auditory areas, all implicated when doing an interpreting task. So what can we conclude from this? Not a huge amount, other than using two languages at once requires most of the language network. Good. Okay, so this was the basis from which I was able to do a little bit more, or go a little bit further myself, which was a few years later. And I then tried to pick up where these pioneers had gone. So I was inspired by them for the tasks that we used, and by the people in Geneva who were the trainers of interpreters. And there we decided to really go in to look better. All of these previous studies have been very small amounts of participants, and we were also interested in the development of expertise. So we started at the sort of beginning, if you like, and we recruited trainee interpreters to look at this. We recruited control participants as well. And what this enabled us to do is to really look at, what happens in the naive case, and ultimately we looked at these trainee interpreters through the training programs, so over the course of a master's in conference interpreting. So we tried to pull apart this notion of what is the control and what is the expertise. We didn't manage necessarily to do that perfectly, but we got some of the way. So here's what we did, and here's how we found it. So this is my MRI scanner. I have neglected to mention that MR scanning is very, very noisy. It sounds like it's being sent over there. Let's see if I can give you another demonstration. All right, anyone who's had an MRI scan for clinical reasons knows what this is like. It's horrible. So this is a very, very noisy technique, and I'm about to ask people to do an interpreting task while it goes, which seems a little mean. So we had to develop a technique that allows us to do this, and what we did was to take advantage of a particular feature of the brain signal that you get in MRI, which is that because we're not measuring the neural activity, but rather the metabolic activity, that metabolic activity lags behind the neural activity. So the brain does something, the neurons fire, but only then are they hungry for blood, if you like. And at that point, the signal emerges. And luckily for us, that signal peaks around 5 to 7 seconds after the stimulus. So we were able to just ask people to do a task during silent breaks, then to take a scan, then to have a silent break, take a scan, etc., in what's called a sparse imaging mode. So this is a benefit of the fact that we have poor temporal resolution in MRI, so be it. It enabled us to ask people to do these tasks, to shadow, so basically just repeat what they heard in the source language, monolingual task. We emphasize to them, unlike a real interpreting scenario, we emphasize to them that we want them to respond quickly and to track as closely in time what they're doing. So this is our monolingual control task. And we have an interpreting task where we ask them to listen in the same source language and to produce the target language, which was their L1. So we're going for a fairly traditional interpreting mode where people go from a less well-mastered language to the one that they consider to be their best language. And we also had a listening task, which is very, very boring. We told them not to say anything, just listen. And we arranged this in such a way that they had blocks. They'd hear 4 sentences, one after the other. So it's sentence, scan, where they do a particular task. And the sentences themselves were arranged in such a way that they would build up a little story, which attempted to mimic what happens in interpreting, which is the interpreter gets more and more information with which to anticipate. Obviously, with 4 sentences, it's minimal, but we tried to mimic that as best we could. And then, having recruited some very patient and very talented participants, a total of 50, we were able to look at the brain signals that we were recording while people were doing these 3 tasks. And I'm going to show you as much as I can of these results. So firstly, I want to talk about just shadowing. So this is the monolingual task where you're required to repeat back what you hear within language as quickly as possible. This is a task, therefore, that is composed of listening, perception, production, self-monitoring, and potentially some incidental comprehension, but they're not required to comprehend. So what do we see? I've presented this in a way that I hope is clearish. This is the left hemisphere here. This is the right hemisphere. And what these are slices taken through the brain. So on the left column, here we're in the more or less the middle of the brain. As we go to the top, we're further out. And this is the same for the right hemisphere. And here, this is a slice taken through the middle this way. All the blue areas are those that are significantly engaged when people are shadowing, significantly more than when they are just listening to speech. So this is everything in the brain that is responsible for or is excited about speaking and listening simultaneously, more so than just listening. And we find areas that are very predictable, very intuitive. Here we have a huge mass of bilateral motor areas, including Broca's area, so speech production areas and motor control. This is something we need when we produce speech. Here we have Wernicke's area, so it's responding when people are listening to speech, and it's a bit more responsive when people are listening and producing speech than when they're just listening passively. Potentially that reflects something to do with comprehension, but hard to know. We didn't ask them how much they understood or retained. So here I'm just trying to put these into 3D context. Now we're scrolling through to the middle of the brain. And here, these midline areas, I mentioned from the Rinner and colleagues study this idea of the supplementary motor area, a motor area it's clearly a motor area by name, but it's interested in complex motor planning. That's here, and then we have this extension into an area called the anterior cingulate cortex. It's all jargon, I'm sorry, but the anterior cingulate cortex is well known to be involved in self-monitoring and error detection. And this is a demand of this shadowing task, even though, you can do it. We didn't score them. We didn't give them any points. Nevertheless, when given a task, people are remarkably compliant. And clearly, if you want to shadow, then self-monitoring is very important. There's also a huge amount of activity going on here in the cerebellum, which could be put down to just being motor control and fine motor control for speech. Okay, but this is just a monolingual task, and that's fun, but that's not what we're here for. What's happening in the bilingual case? In the bilingual case, the interpreting task which is a very unnatural interpreting task, I absolutely accept. That which is in red here is everything which is additionally engaged during simultaneous interpreting compared to shadowing. Everything that is involved in shadowing, all the blue, that's also involved in interpreting. But the additional, what happens, what changes when it becomes a bilingual task is what's in red. And what's fascinating to me about this is that what we essentially have is a left lateralized, mostly left lateralized network, which is just shifted a bit forwards compared to the blue, which is really intriguing because there is this property. I mentioned how the frontal lobe is this sort of human, special, cognitive, fancy architecture. Well, there's a general thing, if you like, a general principle of the brain, which is that when tasks get harder, their representations shift anteriorly. And this is beautifully reflected in this here. And what we have here is the pre-SMA, for example, the part of the supplementary motor area, which is anterior to the rest of the SMA. This is also involved in not just motor planning, but also things like task switching and inhibition. And it does this alongside these red areas here, which are next to Broca's area. So this is a network which is really intriguing because we've already covered most of the language stuff in the shadowing task. We've got more now, which isn't really language specific at all. This is additional cortex that's recruited to do this task. See, Broca's area is here. This is some motor cortex. This is some premotor cortex. This isn't language specific. This is motor areas. This is control areas. And similarly down there for the pre-SMA. So what's happening? What does this mean? My interpretation of all of this, including this subcortical activity, which is here, this is not a language area. I'm going to talk about this area quite a lot. This is an area, set of nuclei in the brain, known as the dorsal striatum. And the particular structure that's implicated here is the caudate nucleus, so-called because it has a long tail, so got a cauda. And this caudate nucleus is not an area that is a language area. It's not even really that traditionally associated with language processes. Its usual set of functions are things like this. They're learning, action selection, motivation, inhibition. So what's it doing? For me, the obvious thing is that when using two languages simultaneously in the interpreting context, there has to be selection, there has to be switching, there has to be inhibition of one and activation of the other. And this would be an ideal structure for doing that. That's what it's good at in general in action control. So this was a story that I came up with, and I stand by it. I'm sure that's what's going on in this population. But these are naive interpreters. They don't have training. This is the very, very beginning of their master's course. And there's 27 non-trainee multilingual individuals in there. But still, this story made sense to me. And so I went a bit further and I said, well, okay, if this is about control, then there must be more and different language control if the participant is responding very, very quickly and there's overlap between the two languages for longer versus if they lag and there's less overlap between the source and the target streams. And so this was something I was able to analyze in the same data because we'd recorded our participants and we tried to investigate really whether there were parts of the brain that were more or less engaged as a function of the overlap between the incoming stream and the output. And we did manage to find something exciting. So here, mapped on the brain, this is now auditory cortex, left and right. Sorry, these brains are seen a bit rotated. I hope they're clear enough. For both the interpreting case and the shadowing case, so the bilingual and the monolingual case, and the purple is where you see great overlap. So this is areas that show significant modulation by overlap. So these are the ones where there's a significant correlation between the duration of overlap between the incoming stream and the outgoing stream for both these tasks. And then we look deeper inside the brain, we find that more than auditory cortex, excuse me, we find specifically for interpreting these two structures here, which are the putamen, which are also parts of the dorsal striatum, again implicated in control, particularly the habitual control of action, the low-level control of action, compared to the caudate, which is more involved in cognitive control of action, which kind of makes sense because we're looking really here at the correlation between the mechanics of speaking while there's another language going on. So we assign this to being the sort of low-level control you need to produce the correct language, but using the right phonological set, even though another one is active because you're actually attending to another language. In the auditory areas, we find really no difference, which is reassuring. So this is functionally segregated. So something specific about the bilingual part of the task and the bilingual control requirement is again in this deep brain structure that is, I repeat, not specialized for language. So the summary of all this is this lovely bilateral shadowing network with a left lateralized, anteriorly shifted and subcortical interpreting network. And I've told you what I think about it, that these deep brain structures are heavily implicated in the control requirements of the task. There's something about the bilingual nature of it that's really specific there. But that's what I think at this point, after this work is published. We then went on to look at what happens with training. So our trainee interpreters came back for another scan. Our non-trainees who were doing other courses of study not involving language then also came back, and we were able to compare how their brains dealt with the task after 15 months. So unfortunately, we lost a few of our interpreters on the way. They were unwilling to come back or failed the course halfway through. This happens, there's always attrition, unfortunately, in longitudinal studies. But those that did come back, what did we find? We compared pre-training and then at follow-up, we found that there was a change in the right caudate nucleus in terms of how activated that area was during the interpreting task, that change was specific to the interpreting condition and not the shadowing condition, and it was specific to the trainees. And what was it? It was a significant decrease in the involvement of this area in the interpreters after training, which was not present in the controls. So it wasn't just a task retest effect. It was something about learning to do this task more efficiently. So this, for me, at that moment, confirmed right caudate nucleus, heavily implicated in the control demands of multilingual language juggling. Great. Story's over, right? This is fantastic. I was very happy with myself. And I maintained this position for quite some time, and that was super and, wonderful. I then reviewed the literature with my colleague Laura Babcock, and we looked at all of the work that was out there that looked at anything to do with the brain and interpreting, and we isolated all of the foci that people had talked about. We find this, there's a lot of areas that come up in interpreting. So this is divided into structural studies and functional. So the structural ones, I mentioned this notion that we can try to look at brain structure and fit together, do differences in structure predict abilities, or does experience change brain structure? That's what all the blue ones are, studies that try to do that. And the red ones are studies where people actually looked at the brain interpreting. And if we try to make this a bit more comprehensible on a map of the brain, we can see that a lot of the brain has been associated with interpreting in some studies, but the number of studies is always low. There have not been that many studies, and the convergence is highest here in Broca's area and moderately high in the basal ganglia, which are these dorsal striatal structures implicated in behavioral control, but not really always in language control. So again, hubristically very proud of myself. I've generated a model, I've put everything together in the brain, and mapping the proposed cognitive elements onto a brain network. I'm not going to talk you through the model because what I'm now going to do for the next 10 minutes is tell you how I was wrong. So my goal, and it remains my goal actually, is to try to understand the role of the subcortical structures in language management, because I do genuinely think that they have a critical role, because they are very, very important structures for domain general control. And it is logical to me, if I think of how the brain would have evolved, that structures that can do that would be the ones that would be adapted for doing language switching as well as any other kind of switching. It doesn't make sense to me that some other structure has to be innovated or developed for language control, because language fundamentally emerges from a brain that didn't previously have language, and little by little, incrementally over evolutionary time, acquires this capability, and it must be wired into all of our other action systems. That's my premise. Nevertheless, what I learned from interpreters is a maxim which is absolutely vital, which is always check against delivery. And this, interpreters remind themselves this when they're given a speech, the text of the speech in advance. They can be as well prepared as they want, but if they're just sight translating the speech instead of listening to what the person, the speaker, is saying, then they're not doing interpreting. And this, yeah, check against delivery. What can we do with this? So I want to confirm the language control function of the dorsal striatum. My claim is the caudate nucleus is implicated in controlling the accessibility of lexicosemantic sets. My second claim is the putamen is implicated in the motor control or planning demands of using a language while another is contextually activated. But there are loads of other explanations. It could be just general cognitive load management, or it could be a specific function of unimodal language management. So dealing with the attention, splitting the attention between what you're listening to and speaking. Could be anything, actually. I mean, limited number of alternatives. But so how to test this? You know, if these are core roles in language control, then they should also be seen in another interpreting mode, which is multimodal interpreting. And fortunately, I had the opportunity to ask whether the model generalizes to this other modality. So here, going from this mode, the simultaneous interpreting, unimodal interpreting, so hearing, speaking, to this other mode of auditory manual or visual oral mode. So this is sign language interpreting, which is a common mode of interpreting, but it's very different because suddenly we don't have overlapping channels anymore. And so is there a similarity between cross-modal and unimodal brain networks for this? And I had, I've tried to break it down again. Both need some of the same basics, the buffering, the reformulation, the access to language sets, but it's not the same within channel demands. There's a different kind of interference is possible, different control of attention is required. So which brain regions are engaged in the bimodal context? So this is something I was very, very fortunate to be able to do with the team in Turkey, which was a study using fMRI where we had volunteers who were coders. This is children of deaf adults. So these are children who are born hearing to deaf parents and grow up, therefore, signing and speaking. So they're bilingual from a very early age. Most of the participants here, unsurprisingly, given that they have a lifetime history of language brokering, are professional interpreters and all have done some interpreting. So these are not naive interpreters at all. And it's worth thinking about the difference between these participants and the ones I presented before. Different experiences, okay? These are very different. Nevertheless, yeah, okay, I mean, I think you can all get the general gist. We used very similar methods. Sorry, this is a bit slow. We used exactly the same technique and the same kinds of stimuli in this study as we did in the previous, but we had a few more conditions because we did interpreting in both directions, so from sign to oral and oral to sign. And we also had shadowing in both the gestural and the oral modality. There's some video here that the heroic Idil Demircan, who's the student who worked on this, took from the control room. I don't know if we can see it. This is someone actually shadowing some stimuli in the scanner. That was a very short utterance. And we also have an interpreting video. So I show this because I'm just amazed that it was possible. The participants were incredibly patient, stoic, and did this task. Idil also prepared stimuli such that they could be interpreted and shadowed with minimal articulation above the neck, because people's heads, as you can see, are in the antenna of the MR scanner. So they were just producing gestures around here. Okay, so what do we see in terms of the results? Well, we see Just when we look at passive viewing and passive listening, very straightforward. We see auditory and visual activation. This is extremely reassuring. Nothing too thrilling there. What happens when we make the task a bit more complicated, when we ask people to shadow both sign and speech? So sign is this blue and green map, and speech is this red and yellow map. There's quite extensive activation, and of course they're different because the modalities of input and output are different. So in order to try to get at what's the core of this, I asked what's the intersection of the two networks? And the intersection of these two networks is really, it's intriguing because in principle, you might say, well, why would there be any intersection at all between watching and signing and listening and speaking? Which bit of the brain cares about that? Well, it turns out there is a network, and it's a left perisylvian network, as well as some subcortical areas, and a right area that's quite interesting, that are interested in both of these shadowing tasks, which is really intriguing. And what's extra intriguing was this validation that in the previous work, we'd looked at this shadowing network, which was just unimodal shadowing. And remarkably, although they're not the same, identical, they're very similar. What we reveal here as being common to both types of shadowing and to the previous network of shadowing, which was just unimodal, they're quite similar. So there's some parts of the brain that seem to be implicated in whatever it is that's in a language context about this dual task. And I don't know what that is yet, but I was gratified to see that we'd managed to highlight in our previous work something that was core to shadowing. This was nice. However, when we move to the cross-modal interpreting case, things get a little bit twisty, but it is absolutely fascinating. So again, I'm mapping two things on one brain in different colors, oral to sign interpreting and sign to oral interpreting. And this is a little bit of a mess because obviously in oral to sign interpreting, there is listening to speech, signing, but also monitoring signing. So there's some feedback about the signing. And in the sign oral case, there's watching the sign, and then there's producing speech, but also hearing the speech. So there's a bit of a mess with the modalities. And here again, what I've tried to do in order to simplify this and really get at this question of what is the core of language control, irrespective of the modality, is just to look at the intersection between the networks. And there is an intersection. The fact that there's an intersection is, again, really reassuring. It says there are brain areas that, irrespective of the direction, of the modality, of the cross-modal interpreting, there's something that's core to this. And this is really interesting. But what isn't so gratifying is how little this network differs from the shadowing network. Differs a little bit. But in this population of interpreters, because I'm going to call them interpreters because 16 out of 20 of them are interpreters, who are very experienced, it seems like cross-modal interpreting does not really call upon a great deal more, despite being bilingual, than does shadowing. Even though if we squint carefully and pretend a little bit, we can say, well, this here, SMA, pre-SMA, that's the same distinction as here. And then maybe a little bit over here is a bit the same as over there. Maybe, but we suffer here from having only 19 usable data sets compared to 50 over on the right. We have the difference of expertise. And what we really don't have is something that is definitely, and I'm being cautious here, but we really don't definitely have these subcortical dorsal striatal areas involved in this cross-modal interpreting. So what do I do about this? I have to revise my model. I have to rethink. And that is, oh, something is broken. So this is where I wanted to conclude, which is that we've done the right thing. We've followed up. We've checked against delivery. We've tried to attest whether this was correct. But at least in the case of experienced bimodal bilinguals, executing cross-modal SI does not necessarily show the same subcortical relationships, as does interpreting for naive or less experienced interpreters. And while I can explain that, where I have to go is to say that bilingual control does not always depend on cortico-subcortical networks, at least inexperienced interpreters. And these are all the differences that we need to think about, but the red line is the one that's most important, which is that the characterization of the bilingual experience currently is too simplistic for me to really understand the results of the study. There's so much more that needs to be accounted for and needs to be dealt with. And I'm trying to, I'm going to say, let's find the positive. And the positive is this. There's a mismatch between the cross-modal and the unimodal case. But that means I have to ask more questions. We all have to think about the other questions we have to ask. And here are some. What is the impact of expertise in interpreting? What are the effects of this early versus later bilingual profile? What are the effects of language asymmetry, not just in terms of modality? And what else happens? There are so many other forms of multilingual mediated communication to think about. There's dialogue interpreting, there's sight translation, there's a whole world out there, and my model needs to explain them as well. I criticize the other models for not being able to explain SI, but I made a model that doesn't explain cross-modal SI. So thank you very, very much for listening. I mean, loads of questions if you want. I'm ready to answer all of them. And I really appreciate your patience with the technical issues and so much of the didactic stuff. So thank you. It's been a pleasure.
Yeah, thanks a lot. So that was really fascinating lecture. Thanks a lot, including the introduction. It was all clear.